Hi, everyone. I'm Paul Yip. I'm a clinical biochemist here at Sunnybrook Health Sciences Center and also from the University of Toronto. I'd like to thank the organizers, LabRoots, for organizing the clinical diagnostics virtual event and for inviting me to talk about the role of proficiency testing in achieving global outcomes for hemoglobin A1c. And I also want to mention that November is Diabetes Awareness Month, so this is great timing. Here are my learning objectives. I'm going to state the benefits of an accuracy-based proficiency testing scheme for hemoglobin A1c, and also to understand the traceability of A1C assays to the NGSP and to the IFCC reference methods. So just as a build-up, I am going to describe the burden of diabetes that we're facing as a worldwide epidemic. This is a problem both in developing. In the US alone, there's 30.4 million people, that's 9.4% of the population, that have diabetes. Some of them are diagnosed, but a large number are also undiagnosed. And it's also really significant because 1.3 million new cases came about in 2017, and there's going to be another million and so on after that. In Canada, it affects more than 3.4 Canadians, and a statistic that we like to use here is that more than 20 people are being newly diagnosed with the disease every hour of every day. And if you actually look at the US numbers, that is even bigger. That's like well over 100. So this is a very significant problem. So diabetes, of course, has been defined by blood sugar levels, and blood sugar is the standard because this is what is measured and what the body has difficulty controlling when there is diabetes. And so that has been the mainstay of testing. Now, A1C has come on board over the past little while, and it's a more indirect way of measuring blood sugar. And so here are some of the differences between A1C and blood glucose measurement. So A1C represents an average glucose over the previous 2 to 3 months, whereas blood sugar really is just the immediate number. Now, both values can vary physiologically. Now, the advantage of A1C is that it is less affected by acute stress and also a large number of common drugs. So while there is some physiologic variation, it's not as much as blood glucose levels, which certainly go up and down, certainly throughout the day, before and after meals. Both are predictive of disease, so it is a very useful biomarker in that regard. A1C becomes so prominent? Well, there's no patient preparation required. There's no fasting that needs to be done prior to the test. On top of that, the sample itself is stable, whereas if you were doing a blood sugar measurement, you'd have to make sure that it was collected in the right tube and make sure that there's no breakdown of the sugar. So overall, there is a huge convenience for measuring diabetes, for measuring A1C, as opposed to blood glucose. Now, there are some caveats. We're not measuring the sugar directly. We're measuring sugar that's attached to a hemoglobin molecule. So if there are disorders that have to do with your red blood cells or any diseases that may affect the red cells in your body or the hemoglobin in your body, that could have an impact on A1C measurements. So come onto the spotlight. Well, this goes back to some of the very early trials, such as the DCCT trial. And that showed when A1C levels were kept to a low number, that would reduce the risk of the microvascular complications of diabetes. And so these complications included retinopathy, nephropathy, neuropathy, and albuminuria. So it's not just a black or white number. You can see that there is a scale that as you lower the A1C, this reduces the relative risk of those microvascular complications. And so I put an arrow there for the 7% because this is the common target of treatment for those with diabetes. And you can see that significantly reduces the on to other trials, such as the UKPDS, which was for type 2 diabetes. And so these are the really important clinical trials to which our results for A1C have been established. So these guidelines now include various targets for A1C, right? And so treatment has been the long established use of A1C, but more recently, the diagnostic utility of A1C has been introduced. So the diagnosis of diabetes was introduced into the ADA guidelines in 2010, and that similarly came at the same time as the recommendation for screening using the A1C test. Now, in other parts of the world, such as in Canada, the adoption, soon diagnosis, came into the Canadian recommendations in 2011, and then the screening in 2013. Now, the World Health Organization has recommended the use of A1C for diagnosis, but not as yet for the screening. Now, beneath that table, I've shown some of the targets. So the treatment target has been long established in both type 1 and type 2 diabetes. Now, the diagnostic target applies to type 2 diabetes, and type 2 is actually the much larger proportion of diabetics. About like 90 to 95% of diabetics have type 2, and that's where we have this worldwide epidemic. And so the diagnostic cutoff is at a level of 6.5% or higher. Now, in terms of screening, there are different guidelines that are out there. So, below the diagnostic cutoff, at risk for developing diabetes, or you could be having pre-diabetes, which is an impaired fasting glucose or an impaired glucose tolerance. Okay? And so the ranges vary depending on the national guideline that you're using. But one needs to remember that, if you recall in the previous slide, the risk of diabetic complications actually follows a continuum. So even below the diagnostic threshold of 6.5, there is a risk for future development of diabetes or those diabetic complications. So all of these numbers are very relevant. So the use of A1C for the diagnosis of diabetes is now a universal standard, and that was defined in the 2011 WHO report that recommended the use of A1C. And what I want to highlight in the recommendation here is that it can be used as a diagnostic test for diabetes, provided that stringent quality assurance tests are in place and assays are standardized to criteria aligned to international values. And so this is where the role of proficiency testing has really helped to ensure that the use of A1C in the diagnosis continues to be reliable. So that brings us into the learning objectives. And so the first part, I want to go through the benefits of an accuracy-based proficiency testing scheme for hemoglobin A1C. Right. So why proficiency testing? So for those who are working in a clinical lab environment, and as a part of laboratory accreditation, there is a requirement to perform proficiency testing. So for example, the College of American Pathologists has a lab accreditation program. For those laboratories who follow the ISO 15189 standard for medical laboratories, requirements for quality and competence, in section 5.6.3, laboratories shall perform interlaboratory comparisons. Now, proficiency testing is also indirectly stated within the diabetes guidelines. So in the Canadian guidelines, it says to use a standardized validated assays. In the American guidelines, it says to use a method that is certified by the NGSP. traceable to the DCCT assay. So it doesn't really say proficiency testing outright, but what this does imply is that you've implemented a method that is validated and continues to be reliable throughout the life of the test. And so the way to do that is through proficiency testing. Now, I also am very aware that in the US, there are some upcoming changes to CLIA, and whereby one of the proposed changes is to make A1C a regulated analyte, meaning all laboratories offering the test must participate in an A1C proficiency testing program. And so there's lots of discussion going on in the internet. I won't go into it, but I do encourage you to take a look. But I hope you will find from today's presentation that an accuracy-based PT program is going to be vital to provide that rigorous quality assurance program around your A1C testing. So what's so important about proficiency testing? This goes beyond merely just an exchange of samples between laboratories. There are very important rules around proficiency testing. It formalizes the comparison of results between laboratories. It consists of scheduled challenges. It also consists of blinded samples. And these are not just blinded, but they're not to be communicated. So this is truly what it is like to test a patient sample. PT also allows for clinically relevant concentrations so that you're testing the entire range of your assay. It should mimic patient samples in terms of how they're handled. Ideally, it checks the entire process, so not just the analytical component, but if possible, the pre and post-analytical steps as well. There's predetermined performance criteria. So there are limits to which the results must agree among all the different participants. Get your results both against other users of the same test, but you can also look for trends that might be happening over time. Now, if a laboratory were outside of those performance limits, this is where correction or corrective actions would be necessary. So this is actually key to a quality assurance program. So what are some of the goals for A1C testing? I'll break this down to two parts. The first is in precision. And so quite some time ago, quality goals were specified by the ADA, the AACC, and the NACB, such that the imprecision within a lab and between labs would be fairly tight. And so an interlaboratory goal for imprecision would be a CV of less than 10, and your own laboratory's goal should be a CV of less than 2%. Now, they had also defined an all-method goal of a CV less than 3.5%. And this took quite some time to accomplish. And a CV of less than 3.5 was first done in 2016. And probably it was last year in 2018 when it was consistently observed in multiple samples from the PT programs. So now that imprecision has been significantly improved over time, what about the total allowable error? So the current standard on proficiency testing is that a method can achieve a total allowable error of 6% using a accuracy-based grading scheme, right? And actually, we'll be changing this to 5% in 2020. Okay? Manufacturers are already certifying their instruments in order to meet this 5% total allowable error goal. So before we go into specific aspects of PT, let's do a quick refresher of exactly what is hemoglobin A1c. And so glucose that's in circulation enters our red blood cells. This is taken up by the red blood cells, and it can attach itself to the hemoglobin molecule. So on the left-hand side, this is illustrated, by the uptake of the glucose, and on the right-hand side, I've shown to you the molecule attaches itself to one of the chains. Okay? In the first step, there is a reversible reaction, and then in the second step, there's an irreversible reaction. And so hemoglobin A1c is the stable glucose adduct of the N-terminal valine of the beta chain. So what's important here is that you can see how the structure of A1c changes, but that N-terminal amine, which would have been positively charged, now has been neutralized because of the glucose on it. So these are very important things to consider, given that there are many different assays to measure hemoglobin A1c. So A1c is measured by a number of different approaches. I've grouped them here according to the principle. And so methods that are based on the charge separation of hemoglobin A1c include ion exchange, HPLC, capillary electrophoresis, and mass spectrometry. On the other hand, there are methods that separate the different hemoglobins based on structure. And so these include affinity chromatography, enzymatic assays, and immunoassays. So from this, we have various quantitative methods for measuring A1c. So I also want to remind you about the importance of accuracy and precision when it comes to diagnostic testing. So these are familiar bullseye diagrams, which I hope you've seen before. This is to precision, accuracy, and so when we're talking about precision, we're talking about the agreement between repeated measurements on the same specimen. So here on the left bullseye, you can see that there's good precision here. The points, when measured over and over again, are landing in fairly the same spot. Now, what we really want is an accurate assay. Okay, so accuracy is the agreement between the measured value and the true value. So here in this bullseye, you can see that the different test results are centered around the target. So here, if you took the average of these, you could say that it's reasonably accurate. But because each time it's not precise. Okay. So ideally, the test is both accurate and precise, whereby repeated measurements are landing on the target every time you're doing the test. So this is important when we're considering A1C, but there's also another caveat that we have to keep in mind. We have to make sure that our assays have no interference from other species that are present in the sample, and these may be related or unrelated molecules. So we need specificity as well. And so some assays for A1C are more specific than others. Now, why is precision important? Well, we have to consider the sources of imprecision. And so this can come from analytical variation. So this is the test itself, variation. So these two come together because these contribute to the overall variation of the test. And so what's important when we're looking at the trending of results is that we want to make sure that the test itself can identify a real change value in A1C. So we want to keep the analytical variation as low as possible, so that if we see the difference between two successive results is actually due to a real change physiologically. So in order to minimize analytical variation, the lab needs to control the sources. So we want to make sure that the variation between the instruments and the operators is minimized, between changes in the calibrator and reagent lots. If we have a large laboratory, we have multiple instruments, or perhaps even minimize the variation between those different locations. And certainly for serial monitoring, we want to make sure that these are stable over time. So variation is important for monitoring. It's also important because if we're going to use a diagnostic cutoff, we want to make sure that the error around a particular cutoff is minimized as well. So that's where we want to make sure the tests are accurate. And so in a proficiency testing program, we want to make sure that we have the right target. Now, for the most part, we're used to proficiency testing where we're graded by peer groups. And so peer group assessment can be helpful because it can give us the imprecision of a particular method, and your results may be within your peer group, okay? But that doesn't mean that your results are necessarily going to be accurate. So, for example, we could have target plus or minus some limit. We can have method B. It could be a different target for that peer group with its limit. And there could also be C, D, E, or any number of other peer groups in a survey. But how do we know that all of them are agreeing with each other? Okay? And so if we're going to measure A1C using universal cutoffs, we need a gold standard for our proficiency testing. So another important consideration is the right material. Okay? And when we're considering quality control and what we're doing on the lab, we have made use of QC, but it does come with some limitations. We have QC that is stable, lasts for a long time, and we're able to make sure our method is working correctly. But probably has a different matrix than an actual patient sample. It probably has preservatives in there. It may even be modified. So oftentimes we have like a multi-analyte QC. It may even be subject to manipulation, whereby you might need to freeze it or reconstitute a lyophilized QC material, and so on. So QC material is not ideal for monitoring accuracy. We also set our own QC targets. And so although we might have a very good performing method, we know that there's other variation going on, such as lot changes, operator changes, and so on. And so our QC might be adjusted to reflect those changes. And that might actually make our QC targets wider, okay? Because at the end of the day, we want to make sure that, we have to either go on with the testing or we have to troubleshoot it. And we don't want to set our QC too tight that leads to needless troubleshooting. So for accuracy-based proficiency testing, we actually need something that reflects the right material. And so we would ideally have a sample that you would treat much like a patient sample. Okay? And so in an accuracy-based scheme, it would use a fresh whole blood sample. And this property is called commutability, meaning that when you test it on one system or another system, it's going to behave in the same way. Whereas if you used some other type of material that had been like frozen or lyophilized or somehow treated, even though it might be the same sample, those different modifications could actually lead to different results. You want a comparison to a true reference laboratory. So in this case, we're talking about a laboratory that is certified to actually give a reference value for A1C. right? So here I've given a snapshot of a PT program from the College of American Pathologists. You can actually find this data on the NGSP website, and I don't want to overwhelm you with all these numbers. But you can see here that in this particular survey, there were actually 5 challenge samples, 6, 7, 8, 9, and 10. And you can see that it also gives a summary of the method performance across different assays. So there's assays that I've shown on the left-hand side here. Okay. So PT can actually go beyond just looking at your own result and seeing if you're within the limits. It can actually give you a lot of information about how your particular method is performing in comparison with others. And what's useful in this particular example is that it actually highlights some methods that you might need to keep your eye on. Okay? You might be using this method, you might be considering changing to this method. Okay? And so in the top row, there's a number of grayed out boxes that are showing higher than expected imprecision in comparison to those global goals that I'd shown you in the previous slide. In the lower box, there's another method that actually has a bias, and the bias is particularly alarming, but it is something that might give you a bit of a warning sign, especially since it came up in a couple of the samples. Okay? So PT is actually a very powerful quality indicator. So it can actually kind of signal, some changes that one needs to be aware of and possibly take some proactive steps. So that brings me to the second part of this presentation, and that's to give you a few slides so that you can understand the traceability of A1C assays to the NGSP and to the IFCC reference networks. So the NGSP, formerly known as the National Glycohemoglobin Standardization Program, now it is known as the NGSP. And if you're interested in reading in more detail, there was an excellent review that came out this year in Clinical Chemistry that highlights the work that's been done over the past 20 years. So the role of the NGSP is to ensure that there is traceability to the clinical outcomes of the DCCT trial. So even though that ended back in the 90s, those targets that were set are still being used now. The NGSP also certifies the methods for hemoglobin A1c. It also defines the acceptable performance limits for the manufacturers. It also plays a very important role in proficiency testing by assigning the value, this is the target value, for the different samples in the PT program. And lastly, both the value assignment for PT programs is accuracy graded using fresh whole blood samples. And this is one of the figures taken from that reference, and it really shows how A1C methods have improved substantially from the time the DCCT trial ended to today. And you can see back at the very beginning of the PT program, the methods that are around showed enormous variability. So the dashed line represents the target value set by the NGSP, and you can see that some of the methods didn't even overlap the NGSP target. There was just too much falsely high A1C results. This more on target, and once we get into 2010, when the diagnostic utility of A1C was introduced, there was fairly good agreement. You can see that the methods are now generally in line with the NGSP target, and also the size of the error bars is much tighter. And so in 2010, the allowable error was actually reduced by half, going from 15%, down to 8%. And now, when you fast forward to 2018, this is where we currently have our 6% allowable error goal, and that's going to be further tightened. And you can see how the performance continues to improve. So the IFCC has played an important role in the global standardization of A1C results. So the has allowed the traceability to the trials, but at the time, the reference method was based on a cation exchange chromatography. Now, in terms of the role of IFCC has played an important role in terms of helping to fulfill the EU directive. And that is the traceability of the values assigned to calibrators or control materials must be assured through available reference measurement procedures or reference materials of a higher order. So back in the 80s and 90s, there was no higher order reference material or higher order reference method for A1C. So a higher order reference through 2003. Okay. And now manufacturers actually certify their methods with a calibration that's traceable to the IFCC method. Okay. And in order to convert the IFCC units, which are millimoles per mole, there's an equation that relates the IFCC values back to the NGSP units. So here in North America, we are maintaining the use of NGSP units, but in other parts of Europe, in the other parts of the world, mainly in Europe, some countries have started to adopt the IFCC units of measurement. So that will take some time, but there is ongoing global efforts. Now, in my last slide, there's probably a number of you in the audience thinking about, well, what about? This is where we have to be very considerate of having a reliable quality assurance program. Now, first off, there are many commercially available point-of-care tests for hemoglobin A1c, and they are also certified by the NGSP. But where do we stand on this? And so the different countries have taken on a different approach, but I'll highlight the one that came from the ADA. And so in the ADA Standards of Medical Care for 2019, it did state that although point-of-care A1C assays may be NGSP certified or US FDA approved for diagnosis, proficiency testing is not always mandated for performing the test. And therefore, point-of-care assays approved for diagnostic purposes should only be considered instead licensed to perform high complexity testing. And so what this implies is that it would be done in a accredited laboratory environment and thereby have a proficiency testing program to back it up. If it were completely point of care in the context of the US, where it would be a wave test, then proficiency testing would not be mandated. Okay. So you can see that there's an important role that proficiency testing has in making sure that A1C assays are able to uphold this diagnostic utility. So I'm going to finish off and just summarize what I've presented to you. Okay, so first, proficiency testing for hemoglobin A1c is a vital part of quality assurance and a requirement for laboratory accreditation. Based proficiency testing A1C results to positive outcomes in the detection and treatment of patients with diabetes. Lastly, global efforts to standardize hemoglobin A1C results has enabled and sustained improvements in the consistency of A1C results worldwide. And with that, I have reached the end of my talk. If you are watching on the November 14th, I want to wish you a happy World Diabetes Day. And I also would like you to be aware that November is National Diabetes Awareness Month. I thank you for attention, and certainly you can contact me through my email address, paul.yip@utoronto.ca, if you have any questions. Thank you.